we're starting with the origin of life, and we touched on this in 1406, generally speaking. But as far as timing is concerned, and I don't know that I can give a bunch of background on this without talking about timing. As far as timing is concerned, we say the Earth is around 4.6 billion years old. But has life been around that long? No. My next point that I want to bring up real quick, you're going to see the term hypothesis over and over and over again in this chapter. Why? We have no idea because there's no real concrete evidence. We're just going based on what we have found scientifically, and we can back up. So the term protocells are not actual prokaryotes. These are like a prototype for prokaryotes. These are before prokaryotes came around. And then it shows that about 1.1 billion years later, we start to see signs of life. So the Earth is here for a billion years, and then finally life starts to take form. And that's what we're going to cover in this lecture today, that time period in between and how life initially started. We do know, based on the cell theory, which was covered in 1406, that all cells come from pre-existing cells. But then the question is, well, then how did the first cell get here, right? And we discussed that there's not a thing such as spontaneous generation. Do you remember what spontaneous generation is? Like it's just like, oh, there's life. Or like, oh, I want a tree right here and it just grows. That's not a thing. But what we are going to study is what components came together to start to form these life cells, these little molecules. And what's really silly, I'm going to take you back real quick to the levels of organization. We'll start, and you'll see, we'll start with atoms. And what's a group of atoms make? Molecules. And then what's a group of molecules create? Close. I mean, macromolecules is technically, but what's the word that goes there for the biological levels of organization? Compounds. Compounds. And so, Dorian, you are not wrong for saying macromolecules, so I'll put those here. Do you guys remember what our macromolecules are? Yes. I'm going to write those in a different color. Carbohydrates, lipids, proteins, and nucleic acids. Yes, which is our DNA and RNA. So then, when these compounds / macromolecules come together, do you remember what those form? Organelles. And what are some examples of organelles? Mitochondria is a great start. What else? Golgi is not it's a structure that we study now as a specific organelle. What's the other one when we talk about beginning of time? Chloroplast. The mitochondria was for sure what I was looking for, chloroplast. And Golgi is used in eukaryotic cells, but not at the very beginning. And when we put a bunch of organelles together, mitochondria, chloroplast, the nucleus, and so on, what do we eventually form? Cells, which is where we are going today. Okay? This transition. So, are atoms alive? No. Are molecules alive? No. What about compounds? Carbohydrates, lipids? Are these alive? No. Organelles? No. But when organelles come together, they form cells. So none of these on their own are alive, but as they begin to work together, they bring up the components in which a metabolic function can be sustained. So then we have cells. So this is what it means. Life is not spontaneous, but it can come from non-living aspects. So macromolecules, carbohydrates, lipids, proteins, nucleic acids, those are all non-living. Questions so far? Did you have a question? Good deal. Yes?
The organelles, like, they do stuff.
They do stuff.
How are they not?
So on their own, they're what we call interdependent. So they depend on one another in order to carry out their life functions. Like, for example, the mitochondria produces the oxygen that all the other organelles need. So if the mitochondria wasn't there, the other organelles wouldn't sustain their function, and therefore the cell wouldn't be there. Does that make sense? So good question. Here's a nice little timeline showing kind of how things come together. And there's an experiment I'm going to talk to you about. So here's around the formation of the Earth. Then we have our organic molecules, which are carbohydrates, lipids, proteins, nucleic acids. Those come together, form our protocells, which are not technically prokaryotes yet. Then we get to prokaryotes. And then we start to see, once we get to, when we talk about prokaryotes, cyanobacteria specifically, they introduce oxygen. When oxygen's there, we see all life forms take off. Right here, and you'll see within your notes and your text as well, there's not any oxygen present. So life forms are very limited. So the early Earth, the characteristics of the early Earth. First of all, there was a lot of water. There was a lot of rock. There was a lot of gas, but was there grass? No. Trees, insects, like nothing. And it'll also cover this in the notes in a little bit, but we have the sun. So we have water, we have rock and volcano, and then we have the sun. And because there's no biomass, which means living weight, there's no one to absorb the energy from the sun or utilize that energy from the sun. So during the day, what is the temperature like? Super hot. And then what happens at night when the sun goes down? Super cold. So in let's say it was a 24-hour time scale. I mean, it doesn't necessarily have to be, and there's a lot of speculation on that. But in one day, it was super, super hot, where water was evaporating from these large bodies of water, to at night where it was super cold, freezing temperatures. Can you tell me what happens in Texas when we go from hot to cold really quick in one day? Storms. And so there is quite a bit of evidence that there were crazy, dramatic storms, very severe storms. And that's going to be pulled up into our Oparin and Haldane hypothesis, or Miller-Urey experiment. But the Earth had gravity, which allowed, it was so far from the sun, and you'll see it'll mention this in a little bit, that it was ideal. We weren't too close to the sun where we were so hot we would die, and we weren't so far from the sun that we were so cold we would die. We were in this ideal location where we had a little bit of heat, a little bit of cold, and we were able to eventually sustain life. Because we weren't too close to the sun, we had water. Because we weren't too far from the sun, we had water. Because if we were too far from the sun, that water would technically be ice, because it would be so cold, right? And if we were too close, it wouldn't be there because it would evaporate. Because of this, and again, there's water, but there's no atmospheric oxygen, we start to see the formation of these organic molecules. And I'm going to show you how we recreated this hypothesis. Our atmosphere, we had what we call a reducing atmosphere. We had nitrogen, sulfur, hydrogen. We had these gases, still no oxygen. And eventually, what we start to see is what is referred to as a prebiotic soup. Prebiotic, what does that mean? Before life. So it's super hot, it's super cold. As far as in the mornings and in the evenings, we have this dense, thick fog. We have really severe storms, dramatic as far as their impact, maybe tornadoes, fires, volcanoes erupting. So it kind of like, I think there's a picture. Yeah, like this is the picture your text provides, but like you have like lava and storms and water and rock and a lot of cloudy and fogginess. You can see all the lightning in the back, but there's no life. This is again the hypothesis hypothesized version of the Earth because we have no specific proof that this is how it was. But Miller-Urey Experiment. So we said we have this Earth that has no life on it. How did we go from no life to life? Miller-Urey put together an experiment, and it's going to be diagrammed on the next slide, to which they believe led to the formation of organic molecules. What they believed was that in the water, when it boiled, got really, really hot, it began to evaporate, and it would cause storms, so there would be lots of lightning. And then that happened during the day and the evening, and then what happened at night? It got really, really cold. And we saw this happening over and over again. And what they hypothesized is that as that hot, super hot water was cooling very quickly, it began to form what they believed were amino acids. And what were amino acids? They are the building blocks for proteins. That is the first hypothesized organic molecule to take place, or to take form, not take place, sorry. Let me show you their experiment. I'm going to show you how they recreated it, and I'll come right back for alternative hypotheses. So this is their setup. What they did, and you can watch YouTube. There's an animation here, but you can also watch YouTube. There's a lot of people who've recreated this just for teaching purposes, so it may help you understand it a little bit better if you don't when I'm done explaining it. But we have water in here that we're exposing to a flame so that it begins to boil. When that water begins to boil, it will then break up and evaporate. So then it gets forced here to what we call the storm chamber. What's happening in that storm chamber? There's a bunch of sparks. They're putting electricity in there, which is supposed to simulate what? The lightning that was happening on a regular basis. So we have those extreme storms. We have the electricity that's brought in. And then right here we have a condenser. This condenser cools it very quickly. So it goes from evaporating to the storm, and it is exposed to all the electricity, the lightning. And then here we cool it down, and we start to see that these gases start to form clumps inside the water. And right down here is this holding, this trap type setting, where whenever we see mostly water, but then we see small little particles. When they began to analyze those particles, they had the components of amino acids. And this is how they hypothesized our first compounds or macromolecules were formed. Because the early Earth's environment, or atmosphere, not let me say environment, atmosphere led to these changes because of the temperature fluctuations and the atmospheric pressure. But again, look at what gas is not present. Let me say this one more time. What gas is not present? Oxygen. So we don't have everything that you and I are talking about right now. We have methane. We have nitrogen. We have, when we say water, we have the water, that's what's evaporating, and eventually we will hydrolyze it and we'll have oxygen, but that'll be through photosynthesis. Hydrogen gas. We have, and there's also sulfide gas that's in here. It's not shown here, sulfur. But anyways, let me go back. That's their experiment. Alternative hypothesis. So it talked about just showing that with the Miller-Urey experiment, but what are hydrothermal vents? Where are they located? Underground, specifically in the ocean. These are the volcanoes located at the base of the ocean floor. And when those volcanoes erupt, the temperatures are really, really hot. But what are the temperatures normally at the ocean floor? Really cold. So we have those same conditions present there. Perhaps that's what led to the formation of the first compounds / macromolecules. Okay? Again, these are all hypotheses because we're not exactly sure. Yes? All right, so when we start talking about where our cells came from or how our first cells came about, we know, and we studied in 1406, that the first forms of life were prokaryotic cells. But again, before prokaryotic cells came are protocells, which are like a prototype for those. Then, as time passed, and this is the intro slide, we went from prokaryotic to eukaryotic. So, how did we get those first organic molecules? And we discussed the Miller-Urey experiment. The molecules themselves are not alive, but when you start to put them together, they begin to form organelles, and those organelles will eventually lead to life. Do you remember what process is necessary to bring two small units together? Hydrolyzation adds water and it breaks bonds. Dehydration synthesis, and look what's right here. Macromolecule formation by subunits. The evaporation of water, it concentrates these subunits. So we had amino acid. We bring it together. It says evaporate, but what is that really? Taking away the water? Yes. And so dehydration synthesis connects these subunits by removing an H, a hydrogen, and a hydroxyl group. So what is this together? H2O. So we technically studied this already, but I'm just letting you know how this kind of comes together. Okay? Now, what other aspects of the environment helped facilitate this transition? The fact that most of the ground was rock and clay. So the clay was very dehydrated. There wasn't a lot of water in it, and it led to the compilation of these macromolecules coming together. Okay? But the only aspect that's missing from clay is water. So we might be able to get these complex macromolecules or organic molecules built, but what we're missing is that biological membrane. Do you remember what the biological membrane is called? Phospholipid bilayer. And recall if you can, when we initially discussed phospholipids, I said you could take phospholipids and lie it on this table and it would do nothing. Phospholipids don't function until they are in water. And I mentioned, whenever I reference this, that movie Big Hero 6, those magnets. And immediately, whenever it gave it a thought, those magnets would construct whatever it was supposed to construct. When you put phospholipids in water, they immediately form a bilayer, which leads to the formation of a vesicle. Do you recall that term, vesicle? So we have the formation of a vesicle, because we're now in water, and within that vesicle we start to have our organic molecules. And this slowly leads, we don't have a cell just yet, we have a protocell. So we have our phospholipid bilayer showing up because we have water, and we have our macromolecules inside. So you can see that we're inching our way through thousands and hundreds of thousands of years to millions to billions of years to an actual cell. So you can kind of see this progression, okay? And it talks about that there. So we have these vesicles, and they have their macromolecules inside. But in order to be considered a living organism, one of the very first characteristics is that you have to have metabolic function. You have to have energy. And do you remember how you and I classify or quantify energy? ATP. ATP. Now, was there oxygen? Not in the early atmosphere. Not in the early atmosphere. So their efficiency as far as making energy was very low. It's what we would call anaerobic respiration. And going back to cellular respiration, remember in cellular respiration we could get 36 to 38 ATP, but in anaerobic respiration only get 2. So there was a lot less energy. But nonetheless, we're starting this whole stepwise of metabolic function. So here we start to get some energy. Another characteristic of all living things is that they have molecular data. What do I mean by that? DNA. We don't know how DNA first got here. There are quite a few hypotheses. The two most popular are the RNA first hypothesis. It is believed initially that because DNA is double-stranded and it is such a large molecule that it's too complex to have just been here. That perhaps RNA came first. And RNA is just single-stranded. Remember, it's much smaller, it's more simple. So they believe that perhaps, one hypothesis is that perhaps RNA came first, that we had ribozymes, which are the enzymes that work within ribosomes, which is within the nucleolus, if we're talking about a eukaryotic cell, but just to kind of connect all that. That RNA was formed, and then that RNA was then turned into DNA, which allowed for that cell to move on and have its genetic material. So remember, ribosomes are responsible for making proteins. Ribosomes are the site of protein synthesis. So that's where this comes from. We used RNA, like messenger RNA, transfer RNA, and we took that information and we changed it into proteins. That makes sense. That hypothesis worked. Another hypothesis is that we started off with proteins. Excuse me, I just sweat. We started, because we had amino acids. Those amino acids came together. We formed a protein. And then that protein was then read and kind of decoded into DNA and RNA. Both are legitimate. Do we know which one is the actual? The one that gets the most support, though, is the RNA first. The RNA first. The reason why is because at the end of the day, RNA is much more simple than all of this. And we call it a principle of parsimony, which says that we take the easiest route. It will probably be the most logical. Okay, for the first prokaryotic cells. And the only thing on Earth that's prokaryotic is what? Bacteria. So the first life on Earth is bacteria. It's not trees, it's not plants, it's not newts. It's bacteria. Here are the characteristics. We have a membrane, so that's our cell membrane, our phospholipid bilayer. We have a nuclear region. Why does it say region and not a nucleus? Because it's a bacteria and it does not have a nucleus. We have a region with cytoplasm that allows for RNA to be translated into proteins. That's where protein synthesis takes place. We have a cytoplasmic region where energy is formed. Because do bacteria have mitochondria? No. Do you remember how they make ATP? Phospholipid bilayer, yes, in their cell membrane. Bacteria make their ATP using their phospholipid bilayer, their cell membrane. So here's the basic characteristics. So yeah, we have something living, but in order to propagate that species, it has to be able to reproduce, successfully reproduce. So now we're going from protocells to our first prokaryotes. They believe that the earliest type of photosynthesis was not actually using carbon dioxide and oxygen. It was using hydrogen sulfide because, remember, there was no oxygen in the atmosphere. But as these very first bacteria began to carry out metabolic functions and water also hydrolyzing and oxygen being released, because there was more water than anything else, organisms that could use water or oxygen to produce energy were selected for. What does that mean? They survived long enough to reproduce. However, we still have a whole domain, I just said that, a whole domain of bacteria that don't necessarily need oxygen. So did all of these die off, the ones that didn't use oxygen? No, that's our archaea bacteria, the ones that can survive in extreme conditions. So the organisms that could carry out photosynthesis and release oxygen became more numerous. So then we start to see traces of oxygen in our atmosphere. And I want to say, and the rest is history, because then it's just like life just starts to form. But we have stromatolites. And some of you may have seen these before. You would have to travel outside of the state of Texas. But these are mineral deposits left from the very first cyanobacteria. So mineral deposits, they look like rocks, and this is actually in a conservation area, but they're estimated to be around 2,000 years old. This is what we use as fossil evidence of the first cyanobacteria and their ability to carry out photosynthesis.
You go into that Stromatolite slide.
This one, yes. You got a question? Yes, huh?
Yeah.
You do have a question?
No, I mean, like,
Oh.
I think you said, did I get it?
Oh, yes, sir. I was like, Sorry. All right, so we have prokaryotes, which are bacteria. So how do we get from bacteria to me and you? The endosymbiotic theory. So for those of you who haven't partied with me before, I like to dissect words. So like endo tells me what? Inside. And symbiont or symbiosis is it's a relationship between two organisms. And throwback, but there are 4 types of relationships. It can be a mutualistic relationship where we both benefit. It can be a commensalistic relationship where I benefit, but you're not harmed. I'm just the only one gaining from our relationship. It can be a parasitic relationship where I benefit and you are harmed, or it can be a predatory relationship where I benefit and you are dead. Those are the choices, okay? So when we say endosymbiotic or endosymbiont or endosymbiosis, we're talking about one organism living inside of another and having one of those 4 symbiotic relationships. Now, do you remember how one of them got inside the other one? It was engulfed. I love that you said the word engulfed. I love that. Because engulfed tells us that it was not eaten. And I used this example in 1406, but how I just said the earth was fluctuating. It was super hot, super cold. There were storms, and we're all bacteria. But I'm clearly bigger than you. Okay, and so I'm a bigger bacteria, and I just kind of bump into you and kind of engulf you. And I'm like, I don't know what happened. And you're like, hey, I'm inside. I'm like, I don't know how you got there, but I don't know how to get you out. But how do we know that they were engulfed and not consumed? They're still there. They're still there. Okay, so they believe, and there's quite a bit of evidence, and I'll discuss that mitochondria and chloroplasts are remnants of prokaryotes that are found in eukaryotic organisms. Do you remember what characteristics of mitochondria and chloroplasts, what they have that tells us that they were once bacteria? They have 2 plasma membranes, or 2. They have circular DNA, where you and I have linear. They have 1 circle chromosome. Do you remember how they reproduce? Not by mitosis. Starts with the B. Binary fission. Binary fission. Those are all indications to us that mitochondria and chloroplasts were once prokaryotes, and they just accidentally got engulfed. So there's a lot of hypotheses, and this also gives us evidence that we shared a common ancestor. And who's our common ancestor? Bacteria. Okay, so it talks about that eukaryotes developed from a symbiotic relationship between prokaryotes. So they accidentally engulfed one another. And it wasn't just a single isolated case where one was engulfed. It started to happen over and over again. Because if it was a single isolated case, we would not be here. It had to have kept happening over and over again. If you were not a photosynthetic prokaryote, so if you're photosynthetic, what are you capable of doing? I can use chemicals or sunlight, whatever it is, to make my own food. But if I'm not photosynthetic, what does that mean? I have to eat other things. I have to consume things that have the nutrients and energy that I need. Aerobes fully exploited energy. What is an aerobe? Something like you and I who use oxygen. They carry out that process called cellular respiration. And we're selected for because our energy levels are higher. So we can compete better. Do you see where I'm going with all that? Well, if you're an anaerobe, you're on the downslide because there's oxygen in the environment, and it mentions here predatory anaerobes, but there's also facultative anaerobes and obligatory anaerobes, bless you. If you're an anaerobe, you function best with no oxygen. But because you have no oxygen, your energy levels are lower, right? So what we start to see is these predatory anaerobes, and it mentions that they eat the aerobes at first. Okay, well, you have a whole bunch of energy, right? Because you're an aerobe. I'm going to go ahead and eat you so that I get the energy that you get. What's the problem with that? It doesn't completely transfer. That's just a quick fix. What has to happen in order for that anaerobe to become more successful? Instead of eating that aerobe, it should probably do what? Work together. It says that we begin to see this symbiotic relationship between the predatory anaerobe and the aerobe when the aerobe is not consumed. So that means when the anaerobe says, I'm just going to engulf you, and I'm going to let you do all the work. You have to stay here. But because you're here, I will eat for us. I will make sure you have the food you need. I just need you to make my energy. I need you to function and give me higher levels of energy. So that's that symbiotic relationship there. Because they're both benefiting, lest one was kind of taken prisoner, but because they're both benefiting, what type of symbiotic relationship is that? Mutualistic, right? So then we begin to see, of course we see that, then we begin to see that the cell membrane begins to, the term is going to be called invaginate, and it'll say that. It just folds in. And that's where we get the Golgi apparatus. That's where we get the endoplasmic reticulum. That's where we get the nuclear envelope. That's where we get all of that stuff. When the cell membrane begins to fold in. The infolding of the plasma membrane, also referred to as endocytosis. That's what forms the endomembrane system. And let me show you what I'm talking about here. So here was our first prokaryotic cell. We see here that we begin to engulf aerobic bacteria. Anybody want to hypothesize which bacteria this is? That we have evidence was engulfed? Mitochondria. And then it continues, and here we have the invagination or the folding, and you can see it's referenced as invaginations here, the folding of that membrane, so we start to see membrane systems form. And then over here I have cells with just mitochondria, and over here I have cells that are beginning to engulf photosynthetic bacteria. On the timeline of life through the endosymbiotic theory, which was engulfed first, the mitochondria or the chloroplast? There's direct evidence that the mitochondria were engulfed first because every eukaryotic cell has mitochondria. Every eukaryotic cell has mitochondria, and that's how this is stepwise presented. That the mitochondria were engulfed first. We got a little bit more maturation going on. And then a lot of them survived with just mitochondria, like you and I, but some of them also engulfed chloroplasts, and these are the ones that became autotrophic. Okay? So scientific evidence shows that the first prokaryote to be engulfed and successfully maintain a symbiotic relationship was indeed the mitochondria, and the chloroplast came next. I'm going to go back a slide. So the final step, I engulfed you, and we have this relationship where we're working together in order to make this permanent so that my offspring and future generations will also benefit from this agreement that you and I have. We need to have something in writing. And whenever you're a living organism, where do we put it if we need it in writing? In your DNA. In order for my kids to get it, it has to be in my DNA. And that's what it tells you here. The very last step is gene transfer. You and I had an agreement, and in order for that agreement to carry on throughout our future generations, we need something in writing. So I need a piece of your genetic material to include in my genetic code so that I can guarantee that all of my future offspring will have this same setup. And because when gene transfer takes place and it becomes part of my genetic code, we create not this symbiotic relationship, but what we call an interdependent relationship at this point. What does that mean? Interdependent. They need each other. We don't just have an agreement. If we're not together, we cannot sustain life. Okay, so once those genes were transferred and it becomes part of the genetic material, that's set in stone. Well, technically set in DNA. And it's propagated. It's talking about specifically right here, the mitochondria, but we see the exact same thing happening with the chloroplast. Okay, so we know, and we said this in 1406, that the mitochondria and chloroplast are both excellent evidence for evolution. Okay, through the endosymbiotic theory. Questions so far? Okay, so this is showing you the invagination patterns, or the endocytosis, or the infolding of the plasma membrane, so how that endomembrane system was formed. Again, we have quite a bit of evidence that backs up that mitochondria and chloroplasts were once prokaryotes that were brought into these symbiotic relationships. Okay. Here is this, first of all, is this organism prokaryotic or eukaryotic? It's eukaryotic. You may not even know the name of this organism, but what tells you that it's eukaryotic? You can see different things. Clearly there's a nucleus, but you can see different shapes in it. And a bacteria, can you see all that? No. So just using, like, my favorite thing to say is, can you navigate or can you orient a diagram? Can you tell me what you're looking at? No, maybe not exactly, but I do know that that's eukaryotic. I don't know exactly, I can't give you the species or anything like that, but this is a protist, and this is us showing that it's got chloroplasts that are crazy similar to cyanobacteria. And cyanobacteria would be the makings of chloroplast in the past anyway. So this is a protist, okay? So Archaeans and Eukaryotes. So bacteria, archaea, and eukarya are the three domains. And we studied those in 06. Bacteria, archaea, and eukarya. What distinguishes, bless you, what distinguishes regular bacteria from archaebacteria? Archaebacteria has to live in extreme environments because it is lacking peptidoglycan in its cell wall. And we kind of covered that, but it's lacking peptidoglycan in its cell wall, which makes it extremely vulnerable. So it can only survive in environments where nothing else can access it or harm it. But when we analyze the genetic material of bacteria, archaea, and eukarya, the archaea are more genetically similar to us than they are to bacteria. Do you remember what these are? Right, those are the aspects that we splice out when we're going through protein synthesis. We have our introns and exons. Exons are expressed. Introns are removed. Exons are expressed, introns are removed. Guess what you and I have? Introns and exons. Guess what regular bacteria just have? Exons. So when we see this organism that have introns, we're like, oh, there's another evolutionary link that kind of shows us this progression into eukaryotes.
So basically, like, are the archaeobacteria like the organelles that weren't necessarily became eukaryotes? They kind of like just split apart, kind of.
So you just said organelles became. I know that's became.
Not necessarily were close enough to become eukaryotes, but just kind of became their own thing.
They are their own thing. Yes, there was a genetic mutation at some point, hypothetically, because we don't know exactly, but that where the carbohydrate peptidoglycan was no longer deposited in the cell wall. which when I taught peptidoglycan, I said it was like football pads, and it protected that. And because it didn't have that, in a regular environment like this, us touching it would eliminate it immediately. So it has to live in environments. But so we kind of see with that mutation and that unique genetic setting, but it also leads to the introns, which shows us a connection evolutionarily, time-wise. Does that make sense? So how did we go from bacteria prokaryotes to our eukaryotes? We see our archaea having the introns. We can see this, but we still just had unicellular. So how do we go from being unicellular, which means what? One cell, to being multicellular? Because you and I are multicellular. We're 100 trillion cells. When we look at evidence, the molecular clock, which means I'm looking at what? Molecularly, though. DNA. When I look at DNA and DNA is analyzed, they believe that we start to see this multicellularity show up around 800 to 100 million years ago. But if we look at fossils, the fossils only back us up to 800 million years ago. However, the first multicellular organism didn't show up like this. The first multicellular organism, they believe, was a bunch of unicellular organisms that lived in a colony. What does that mean? They just all lived in a group. So they had this big mass of individuals all living in this one group, this colony. And they continued to do that generation after generation after generation. But guess what began to change? What's the only thing that can cause you to change? Mutations, which are caused, well, that's a random mutation, but what can cause the environment? So we've been successfully living in this colony for a really long time, all just in our group doing our own thing, but the environment begins to change. So then we start to see cellular differentiation. So what that means is all of us come together, let's say we're all individual cells. We're just hanging out, doing our own thing, but the environment begins to change. And over here, it's really, really hot. And over here is where our food source is. So I need these individuals over here to start doing what? Working on protection from the heat. So that's what I need you to focus on. And I need these over here to focus on getting me food. And I'm using a very simplistic example, but that's what starts to happen is because the environment changes. We're living in a colony. We have to start assigning or delegating responsibilities. And as you begin to delegate responsibilities, you end up with this section being an arm, this section being a leg. So all the cells you are started off as one cell. You are one cell. And it started to divide, and then they said, hey, I need you to take care of this. I need you to do this. You become a heart, you become eyes, and so on. That's what this is saying here. The first multicellular eukaryotes were in colonies, and they coordinated activities to adapt to their environment. Eventually, this coordination required gene expression. So genes to be turned on or off. And as a result of that, we start to see the cells change their phenotype. And what does that phenotype mean? What they physically look like. So ultimately, it is believed that just because of where the Earth is as far as distance from the sun, that it was inevitable that life was going to show up at some point. So as things began to change, the environments began to change, and it mentions again the reducing atmosphere, so the gases in the atmosphere, the moderate gravity as a result of distance from the sun, and just the distance from the sun. We're not too close to catch on fire. We're not too far to freeze to death. Ultimately, it led to us having life. And so here it kind of shows that timeline that we discussed in this chapter. And that is it.